Hi everyone. In this lecture we're going to focus on the reproduction of angiosperms or flowering plants. On this slide we have an illustration of just how wide the variety of forms that the seeds of angiosperms can take. In the upper left is the seed from a coco de mer. This is a type of palm tree and it holds the record for the largest seed. The biggest weighed in at about 55 kilograms. Orchid seeds, like the one in the bottom right, are some of the smallest. A poppy seed weighs about 3,000 times more than the smallest orchid seed. In the previous lecture we saw that seeds evolved here, and we've talked about gymnosperms, which is one lineage of seed plants. And now we're going to look at the results of an important evolutionary innovation here, that of flowers. This is a character that is shared by all of angiosperms. In this lecture, we're going to look at how the angiosperms have co-evolved with animals that pollinate them and the flowers that play an important role in this. In gymnosperms, pollination occurs on the ovulate cone. In angiosperms, pollination occurs in their flowers. Most flowers serve as a tool to attract pollinators. Indeed, the efficiency of pollination is increased in plants pollinated by animals compared to those plants that depend entirely on the wind to disperse their pollen. Plants with wind-dispersed pollen have to produce a lot more pollen than those that rely on animal pollinators. As you can see here, with this small sample of flowers, flowers are spectacularly diverse in size, color, scent, and form. However, as we'll discuss on the next slide, they have all the same basic organization. Shown here in the upper left is a lady slipper orchid. Next to that is a magnolia flower from one of my favorite trees, Magnolia grandiflora. In the bottom right, we have the top portion of a French lavender, and in the bottom left, we have a flower from a tropical tree known as the scarlet flame bean. This tree is actually part of the legume family and is related to beans and peas. Flowers are made up of concentric whorls of floral organs. The center two whorls contain the reproductive structures and are made up of ovule-producing carpels surrounded by the pollen-producing stamens. Each flower may produce several carpels, but because they're often fused, it may seem as though there is only one at the center of the flower. Each carpel has an ovary at their base, and this ovary may be one to many ovules that will develop. The ovules are the female gametophyte. The ovary helps to protect the ovules from being eaten or damaged by animals. Because the ovules are enclosed, these plants are called angiosperms, which arise from the Greek for vessel and seed. Gymnosperms, on the other hand, have exposed ovules, leading to their name, which means naked seed. While the ovary protects the ovules in angiosperms, it also makes it impossible for pollen to land directly on the ovule surface. Instead, pollen must take a more indirect route to reach the ovules. In addition to the ovary, the carpels commonly have a cylindrical stalk called the style, with a sticky or feathery surfaced top, which is called the stigma, and this is where pollen lands. The next whorl out and surrounding the carpels are the stamens. Stamens are typically made up of a thin stalk called a filament that supports a structure called an anther. Within the anther, the male gametophyte or pollen develops. In some plants, the stamens are more leaf-like with sporangia on their surface. Surrounding the carpels and stamens, most flowers have two additional outer whorls. These whorls do not produce gametophytes, but they do contribute to reproduction. Just exterior to the stamens are the petals. This whorl is often brightly colored and forms distinct shapes and odors. Petals can serve to attract and orient animal pollinators. The outermost whorl is made up of sepals. Sepals are often green and encase and protect the flower during development. Other sepals may be brightly colored and hard to distinguish from petals. For example, in lilies, the sepals and the petals are not easy to tell apart, and so both are called tepals. In order to recruit animals to aid in pollination, plants must be able to attract them. Flowers communicate their presence to pollinators through the use of scent and color. However, in order for pollinators to be interested and to ensure that the pollinator visits multiple flowers of the same species, there must be a reward for them. In many cases, the reward is in the form of food. Many insects consume some of the pollen, like this butterfly. Nectar, which is a sugar-rich solution, is produced by many flowers to attract animals, as in the case of this hummingbird and bat. In other cases, the reward is not food, but some other resource required by the pollinator. But in other cases, there is no reward at all. Instead, the flower tricks the animal pollinator. Here are two examples where flowers use mimicry to achieve pollination. In the lower right photo, the flower has the smell and appearance of rotting flesh to attract flies. However, when the flies arrive, there is no food for them and they end up pollinating the flower in their search. The species of orchid in this image to the top right has the shape and smell of a female bee. This causes male bees to try and mate with the flowers, taking up and depositing pollen in the process. Let's take a look at the angiosperm life cycle. As mentioned in the previous lecture, a major trend in the evolution of plants is the decreasing size and independence of the gametophyte generation and the increasing prominence of the sporophyte generation. In angiosperms, this trend is taken even further. The male gametophyte has only 3 cells. One controls the growth of the pollen tube, while the other 2 are the male gametes or sperm. In most species, the female gametophyte contains only 8 nuclei, arranged into 6 haploid cells, one of which will develop into the egg, plus a central cell that contains 2 nuclei. The central cell plays an important role in the development of the seed and the support of the embryo, which we'll discuss more in a moment. Angiosperms have a wide range of mating systems. At one extreme, there are self-compatible species, in which pollen and eggs produced by flowers on the same plant can unite and produce viable offspring. These species are able to reproduce even if they are physically isolated from other individuals of the same species, or when pollinators are rare. At the other extreme are species in which pollen must be transferred between different plants. These plants are self-incompatible, meaning that pollination by the same or closely related individual does not lead to fertilization. Such self-recognition is based on proteins produced from the self-incompatibility genes, or S genes. We will first focus on the development of the male gametophyte, which takes place in the anther portion of the stamen. There are typically several anthers present in the stamen whorl. Each anther possesses 2 to 4 sporangia, which we can see here in this cross-section. Within each of the sporangium, there are several diploid cells that undergo meiosis to form haploid spores. A haploid male gametophyte will develop from each of these spores via mitosis. As it develops, it will be surrounded by a sporopollenin wall. Like I mentioned, the mature male gametophyte is comprised of 3 cells, 2 sperm cells and a tube cell, which plays an important role in a moment. Now focusing back on the carpels, here's the development of the ovule or female gametophyte. Similar to the male, this starts with the sporangia. The sporangia will grow a bit before a single cell within the sporangia undergoes meiosis. This will produce 4 haploid spores. However, only one of these spores will go on through several rounds of mitosis to become the multicellular female gametophyte that fills the sporangium. This gametophyte will have 7 cells with 8 nuclei. The other 3 spores will undergo cell death. The next step in the angiosperm life cycle is pollination, followed by fertilization. When the pollen is mature, the anthers rupture, allowing the pollen to be dispersed. A mature pollen grain contains 2 sperm in a pollen tube nucleus. When a mature pollen grain lands on the stigma of the female carpel, the pollen tube nucleus develops into a pollen tube, which grows down the style to an ovule in the female ovary. In this top image, we have pollen that has landed on a corn silk and begun to grow its pollen tube into the style. The large number of pollen grains and the long length of the corn silk help to create competition between the male gametophytes, leading to natural selection. In this bottom image, the pollen tubes have germinated and grown down the style. Many of them look to have entered some of the ovules. As the pollen tube grows, the two sperm cells will travel down the pollen tube and then enter the ovule. One of the sperm will fuse with the egg cell to form a diploid zygote. The second sperm cell will fuse with the two haploid nuclei of the central cell. This will form a triploid cell. During development of the seed, this triploid cell will undergo several rounds of mitotic divisions that will form a new tissue called endosperm. In angiosperm seeds, it is the endosperm that supplies nutrition to the embryo. The process by which two sperm from a single pollen tube fuse with the egg in the central cell is called double fertilization. By double fertilizing, the female gametophyte provides food for the embryo. However, endosperm development only occurs after fertilization. This prevents the plant from investing valuable resources into a seed that has not been fertilized. This differs from gymnosperms. Recall that with the pine, it was the female gametophyte that provided nourishment for the embryo, and this occurs regardless of fertilization. The seeds of many plants consist mainly of the endosperm. The embryo itself is small. When the seed germinates, the embryo will draw resources from the endosperm to help it to become established. The endosperm will provide all of the nutrition for the plant until it can begin photosynthesizing. In some plants, the endosperm is absorbed by the cotyledons during development. The cotyledons are part of the embryo and are considered embryonic leaves, not true leaves. The cotyledons will store the resources from the endosperm and then release them to the embryo. Successful dispersal of seeds are enhanced by the formation of fruit. As the embryo and endosperm develop, so does the fruit tissue. The fruit tissue is predominantly made up of the ovary wall, but it can incorporate other parts of the flowers, such as the sepals. Here on this slide, we have the development of plums. On the left, we have the full flower. Then in the middle, we have the immature green fruit. You can still see some of the remnants of the flower hanging on to the fruit, particularly the stamens. Then, in the right-hand photo, we have the mature fruit. While fruit represents a heavy investment for the plant, they do serve two main functions. Firstly, they protect the immature seeds from being preyed upon by animals before they are able to withstand their dispersor's digestive tract. The tissues of immature fleshy fruits are physically tough and highly astringent for this reason. And secondly, fruits enhance dispersal once the seeds are mature. Fruits rapidly transform in texture, palatability, and color as they ripen. Starch is converted to sugars, and the fruit becomes softer as the connection between cell walls loosen. This increases their attractiveness to animals who will consume the fruit and disperse the seeds within. Animals play an important role in seed dispersal, where they are often attracted by the nutritious flesh of the fruit. In fleshy fruits, the seeds are protected by a hard seed coat, so that after the fruit has been consumed, the seeds can pass unharmed through the animal's digestive tract. In some cases, seeds are dispersed when animals gather more seeds than they can eat. These leftover seeds are then left and can germinate. Other seeds are not consumed at all, but rather have modifications to their outer covering that allow them to catch a ride on animals, such as the cockleburs on this cow. Humans also disperse seeds long distances, sometimes far outside their usual range. There are some angiosperms that do not rely on animals at all for seed dispersal. For example, the down of this milkweed allow it to use the wind to carry its seeds away from the parent plant. The wings of maple seeds do something similar. Other plants use water to carry seeds. For example, coconut can disperse via the ocean, where they will float until they are washed up onto an island. It is not always in the seed's favor to germinate immediately after dispersal. Some plants have evolved seed dormancy, which is an adaptation that delays seeds from germinating during unsuitable ecological conditions that would typically lead to low seedling survival. Some seeds will prevent germination even when the conditions are favorable for them. In some species, dormancy is enforced by hard, tough seed coats. This is also called physical dormancy. The impermeable layer of the seed coat prevents the seed from taking up water or gases that might break dormancy. This type of dormancy can be broken by several factors, including high temperatures, fluctuating temperatures, fire, freezing and thawing, drying, or passage through the digestive tract of animals. These factors weaken the seed coat, allowing the seed to germinate. Some other species, dormancy is regulated by the embryo. In some cases, the seeds are induced to germinate if a temperature threshold is reached. This threshold can be a high temperature. For example, the cocklebur seen here on the slide will not germinate until temperatures reach around 30 degrees Celsius. These tend to be plants that require warm soil for successful seedling establishment. On the other end of the spectrum, some seeds need cold temperatures or cool soils to germinate. Light plays a role in the regulation of seed germination in some plants. These seeds need a period of darkness or light in order to germinate, or they need a specific quality of light. Let's start with this figure of the visible light spectrum. Far red light is at the end of the visible light spectrum and is not used by plants for photosynthesis. The shorter wavelength red light, on the other hand, is an important type of light for photosynthesis. In experiments in the 1950s, lettuce seed germination was found to be stimulated by exposure to red light. Here's a representation of that experiment at the bottom of the slide, while exposure to far red light actually inhibited germination. Now, if we look at this data table, we can see that they also found that it was the order of exposure to the different types of light that was important in determining whether seeds germinated. If the seeds are exposed to just red light, a very high proportion of them germinated. But if they're exposed to red light first and then far red light, there's a much lower percentage of seeds germinating. Then, trying different treatments, they found that what mattered was the last exposure. If the last exposure was to red light, a high proportion would germinate. And if the last exposure was to far red light, a much lower proportion would germinate. The way that plants are able to respond to different types of red light is through the use of pigments. A particular pigment, a photoreversible pigment called Phytochrome, is needed to detect the exposure to both far red and red light. Phytochrome is a photoreceptor, and it switches back and forth in between two stable forms depending on what type of light it is exposed to. Exposure to red light causes the phytochrome to change into a form that absorbs primarily far red light. This form is called PFR. Exposure to far red light, on the other hand, causes phytochrome to change back to the red light absorbing form called PR. This means that the phytochrome can convert from PR to PFR and back to PR depending on what kind of light it is exposed to. Now we know that exposure to red light stimulates seed germination, so we know that the active form of the phytochrome is PFR, which is brought about by red light. So when the phytochrome is in the PFR form, it induces seeds to germinate. In the dark, the active PFR slowly converts back to the inactive PR form. So what that means is that at the end of the day, when most of the phytochrome is in the PFR form, once it gets dark, the PFR phytochrome will begin to convert back to the PR form. Then, when the sun comes up and the phytochromes are exposed to red light, they will convert back to the PFR form throughout the day. The active form of the phytochrome, PFR, precipitates changes in gene expression, which leads to seed germination. What the phytochrome ultimately does is that it allows dormant seeds to respond to the quality of light that is reaching the seed and the presence of overhead plants. As sunlight passes through leaves, the red wavelengths are absorbed by chlorophyll, but the far red wavelengths are not, and they're transmitted through the leaves. The ratio of red to far red light is therefore much higher under an open sky than in the shade. As a result, the proportion of phytochrome that is in the active PFR form is much greater in open environments than in the forest understory. Consequently, a seed in the shade that is not receiving much red light, but mostly far red light, is much less likely to germinate, as its phytochrome is more likely to be present in the inactive PR form. Many plants can also reproduce asexually, meaning that they do not have to undergo the formation and fusion of gametes in order to produce a new individual. Some plants that utilize asexual reproduction produce seeds without fertilization. Other plants have offspring grow out of some part or tissue of the parent plant. The majority of plants that reproduce asexually first grow to a new location and then produce the new plant. This is also called vegetative reproduction. Some plants, like lawn grasses, spread out horizontal stems underground and then allow new upright grass shoots to form away from where the parent plant originally germinated. Other examples of plants that reproduce by this means are strawberries, bamboo, and spider plants. If you have ever grown any of these plants at home, you'll be very familiar with their ability to escape and expand via vegetative reproduction. Woody plants, like trees, can also use vegetative reproduction. An example can be seen when a tree falls over and new shoots form and grow upwards out of the fallen trunk. What I think is one of the neatest examples is found in quaking aspens. This tree has a vast spreading root system that continually sends up new shoots. Each individual stem will only live for less than a couple hundred years, but the plant itself can live for several thousand just by sending up new stems. The largest colony of quaking aspen is found in Utah. It's called Pando, which is from the Latin for I spread. Pando is estimated to be about 80,000 years old and covers about 106 acres. If you have any questions, please let me know via the Q&A discussion board.